Okay. So welcome everyone for the conclusion session of Biochemistry NOC23-CY30. So this course was already there in the NPTEL portal and the exam is coming up in 29th of April 2023. So as the session has already ended till week 12, we have already conducted all the week 12 live sessions. Now this session, this 1 hour special live session is as per the request of students. So in this session, I will try to revise all the concept of biochemistry with the modules that are there in the course. And also, if mostly I will take the questions from you people, whoever have the doubts related to biochemistry in the in the modules. So you can unmute themselves and you can unmute yourself and ask related questions. So basically, for the because this video will be uploaded in the YouTube. So for the YouTube people who will be watching there, so I am Karan Kumar, senior PMRF scholar at IIT Guwahati, and this is the WhatsApp group created for the PhD guidance. So whoever will be watching, they can join this WhatsApp group in the and this link will be given in the YouTube link description. And the consolidated NPTEL sheet including which contains all the recordings of week 1 to 12 are in this link. You can open this and can refer all the different interaction recording which is uploaded on YouTube, and this is my official YouTube channel. You can subscribe to this for regular updates and also to learn more concepts. If you have any doubts related to the video content of the respective lecture, you can comment into the comment box, and then I will answer there. Or there are another means to approach me. You can contact me on my email ID. As this session is ending, this is the last live session from my side. But if you still have any doubts related to the course content or that you are facing any problem, so you can email me or you can post that into the WhatsApp group also. This is my website, as you already know, that related to all the projects that I am conducting and the intensive. If there is any opening, you can check it on my website. So uh, let's come to the module. Let's first revisit all the modules that you have learned in your biochemistry this course. So all the live sessions also have provided here in this live session link, recording link. The first module was on amino acid. In this, so what all the topics you learned in the different modules or the different week that we will discuss in the next slide. So what all the modules you had? You had amino acid, then protein structure week 2. In that you had 2 lectures and 1 live session. The live session link for week 2 is here. Then week 3 and week 4, week 3 was again protein structure continuation. Then week 4 and week 5 were related to the enzymes, their basics and the different enzyme kinetics model. Then this enzyme kinetic totally it completed in the week 6, in which the ma'am also talked about the mechanism of how different enzymes catalyze a reaction, what all the important amino acids involved in the catalyzation of reaction or the binding pocket. So total in week 5 to week 4 to 6, you had total of 6 lectures related to the enzyme and enzyme mechanism. Then week 7 was related to membrane and lipids, in which the most of the questions that come from numerical type of questions that come from the like calculating the charge or calculating the potential between the two membranes, two side of the membrane, what is and the change in the potential, what are the charge if it is given, then what will be the kind of exchange of ions from through the membrane. That all problems we discussed in the week 7. Then week 8 was dedicated to nucleic acid, in which it has total total 3 lectures. Then Week 9 was related to vitamins and coenzymes. Week 10 was carbohydrate. Week 11 was bioenergetics, in which ma'am talked about the various different factors of how the electron transport chain is composed of, what are the different like ratio, and how to calculate the various uh, charge and discharge mechanism. So, and also related to the how much ATP is being produced, and these all things were discussed in bioenergetics. So there were some numerical problems in bioenergetics module. Then last week that we discussed on Friday, on 21st of April, that was related to the metabolism. In this, we discussed glycolysis, then Krebs cycle, then related to how these are being like catabolism and anabolism. Then what are the energy associated with all the pathway? What are the different enzymes associated with that? So these all things were discussed in week 12, which contains a total of three lectures. And here are the live session link that I conducted, and you can go through different videos if you have, if you want to practice, if you want a recording of the problem practice session of the assignment. And also in in most of the video, I had taken up the question from the students and solved that out. So but that was not like many things were included in that. So I have specifically kept this session so that whatever doubt you have, you can ask your doubt by unmuting yourself related to these all modules. Okay, so what all the next thing that you all would like to know, that what are the important topics that might be very much important in case of when you are studying, like when you are studying for focusing for exam or exam point of view, what are the important topics? So let me first go to my uh, this course content. Okay, not this one. Or YouTube channels. Another important announcement that I would like to make: if you want to access any of the, yeah, if you want to access any of the various like PPTs of this, that you can find in the same sheet that I had shared. The sheet is this one. Where is the sheet? Yeah, let me open through. Yeah, you can find that in the problem solving sessions. You go to the problem solving sessions in the portal and log into the portal first. Then if you are logged in, then in the problem solving sessions, you can go there, and in that it will open an Excel sheet. And this Excel sheet link I have provided in my like introduction of these slides. So there also the link I was showing. So all the links, YouTube link is also available here. And if you see, this is the PPT link. Okay, if you open this one, so you can find all the PPTs or handout. Total 12 lectures have been, all the PPTs have been uploaded here. You can go through this link and then access this PPT here, right here. And if you go back to this, if you go one step back, if you go to this folder, in the reference material folder, you can find handout of the PPT merge, and there is one more. Okay, I hope that is also accessible. No. Okay, so this, in this PPT link you can get all the website, and for the reference material I will upload whatever reference material you need, like the textbook or anything, you can refer that also in the same link. Okay, if you go one step back to the folder. Okay, now let us see what all the important topics that we discussed in respective weeks and what are the important thing corresponding to your different sections. Okay, so week one. In week one, the important thing is that you can focus on. Not okay. Let me delete this so that I can write here. Yeah. So what are the important topics in respective sessions? Okay. So for week one, the important thing is that you focus on isoelectric point. Isoelectric point will be very much important in any case of like whichever things you will be focusing, whichever exam you will be focusing. Questions from isoelectric point will be there. Then you can focus on what are the amino acid properties and classification. Amino acid properties and classification, which talks about all the what are the size of the amino acid, charge, polarity, then hydrophobicity, aromaticity. Then you can focus on the peptide bond formation. This is not not this much in like important because in module 2 it will all it these are already there, like the primary and secondary structure of the protein and the peptide bond formation and the various phi and psi angles that are there already in the protein structure module. So in amino acid you focus on the various types of amino acid, then how to calculate the isoelectric point and the properties and classification of amino acids. Other than that, you can also focus on titration curve. How to calculate the PI value, what are the PK and PKB, so titration curve. So this will be important. These three things are important in week one. Now talking about week two, if you talk about week two, in week two the important thing is like when you are studying the protein structure and like protein structure and its morphology, then the important thing is you study what are the various protein secondary structure. What are the types of structures that protein can have? Okay, primary, tertiary, secondary, all the things about it. What are the globular protein? So these things you can focus. Another important thing is Ramachandran plot. Always expect a problem in any competitive exam from the Ramachandran plot. And what in when you read Ramachandran plot, then you have to focus on the phi and psi angles. And then if you are studying the protein structure, like the various types of protein structure, then you also focus on what are the various important interaction that stabilize this kind of structure. Okay, so also talk about that, read about it. Then helical wheel, how to prepare a helical wheel. Normally these kind of questions will not come, but you for clarification purpose you can read about these helical wheels. Okay, so this will be from week 2 and week 3 is also about protein. So let us see what we are discussed in week 3. Okay, so in week 3 that was related to the solving protein thermodynamics. Okay, thermodynamics of protein. So in thermodynamics, you have to study how a protein folds. What are the hydrophobic effect? What are the driving force? Then you can also read protein folding, because these problems will always be there in your exam, like protein folding and denaturation, like which are the different agents which help in the protein folding. And you can also read about here in the like Anfinsen's experiment. You might have you must have remembered that. And then what are the different folding patterns, what are the denaturation and denaturants. Denaturants are those chemicals which are used for unfolding the protein to study its different characteristic. Then Chow Phosphine parameters. Now you can also think about the hydropathy scale of the protein. This is important. Hydropathy scale of protein. Now coming to the enzyme section. So these are the important things that you have to refer for any competitive examination. Now let me duplicate this slide so that we can open all three PPTs together. So lecture number 4, week number 4, week number 5, that was also related to protein, and week number 6, that was also related to the protein. Let's go to week number 4. So in week number 4 you studied. So I will take complete like I will suggest the complete enzyme module. If we talk about the complete enzyme modules, then what are the important things that you must focus on? Kinetics. Okay, the variants of kinetics. Kinetics means Michaelis-Menten kinetics. Then LB plot, Line-Weaver book plot. Then ADFC Hofstee plot. So these ADFC EH plot. So these plots will give you the various different characteristic of enzyme mechanism. Then modes of binding you may study or you might not study, that is fine. But you also need to focus on how enzyme catalyze a reaction. Enzyme catalysis. Enzyme catalysis. Then you can focus on bi-substrate reaction, like ping pong model. Normally you don't have to remember the mathematical formula of ping pong model, but you just know what are the ping pong models, how enzyme how enzyme work in this model. Like ping pong model are the bi-substrate model in which the one substrate binds to the one substrate bind to the enzyme and it help into binding of second substrate. And then like one substrate binds, then chemical reaction happens. First product is released, second substrate is binded to the modified enzyme. Now the second product is released and enzyme is restored in its original form. So that is a ping pong model. Another variant is ordered by model. In ordered by model, well, like you know which like both of the substrate will bind together, then both of the product will bind together. So like that, the fashion will be there. So ping pong model, by ordered by model. Then another thing that you can take from the week 4 is this is the only thing that you have to focus on. And another thing, if you want to read, that is the enzyme classification. There are total 6 class of enzyme. So remember that, and also remember what are their properties. Like if in a class of enzyme, if the enzyme is there, then what will be their function? So enzyme classification is important. Like what is the role of ligases, what is the role of your synthase, what is the role of catalase. So catalase works under which category. So this classification you can read, enzyme classification. What are the role of hydrolases? Okay, so that is from the week 4. Now coming to week 5. In week 5 you learned what are the inhibition and inhibition kinetics. So in this, that was the basic enzyme kinetics, introduction to basic enzyme kinetics. Now in the week 5 you learned what are the inhibitors. Normally lot of questions come from this topic only, like whichever exam you go, because that these are very important in case of chemistry also, like the only little variation and the questions will be same. Okay, so inhibitors you can read, then you can read what are the three different type of inhibition models, like competitive inhibition, non-competitive inhibition, uncompetitive inhibition. Then what are the behavior of plots, how you plot the LV model, like Line-Weaver-Burke plot, and kinetics model in all three different kind of inhibition pattern. What is the feedback inhibition? So type of inhibition, types of inhibition. Am I audible?
Yes, sir.
Okay. Then you can read about enzyme mechanism. So enzyme mechanism is more detailed in week 6. So in week 6, ma'am talked about like she also talked about the hemoglobin and myoglobin binding of oxygen. How why hemoglobin is transporter, however myoglobin act as a storage of oxygen. So these are things. And while I was discussing the enzyme mechanism chapter, like week, there I also talked about how enzyme, like how we get like fainting nature, or why we like kind of strike in case of like when we are climbing, then why when we are climbing and the oxygen concentration is low, then what stress is imposes on our body. And in week 12 we learned, week 11 and week 12 we learned why this is happening, why oxygen deficiency is much much more fatal to our body, to human body. Okay, so these all things we talked in week 11 and 12, where why can somebody answer me why oxygen is important? We have discussed that in the great length in all three lectures.
Sir, sir,
Yes.
partial pressure of oxygen plays an important role in binding with hemoglobin.
Okay. And no, I am talking about, I'm asking why oxygen is important factor. Is my screen visible to all of you? I see somebody else has shared his screen. Is my screen visible?
Yes, sir.
Okay.
Hello.
Okay, so yeah,
Well,
can some
for respiration like ATC cycle, electron transport chain, oxygen is important for ATP formation.
ATP formation? Very good, Abhilash. Actually, that I discussed in the week, like week 11 and also in week 6, that why oxygen is important. Oxygen is important because that is the only thing which help in accepting the electron transport chain. The main role of oxygen is there only. Otherwise, we don't need oxygen. Oxygen is the terminal acceptor of electrons, and when it is accepting the electron, during that time, all the complexes pump proton in the inner mitochondrial space. Okay, in between the in between the outer mitochondrial space and inner mitochondrial space, there is a space where all the protons are uh, segregated. All the protons are there, which are being pumped from inside to outside. Okay, from Christie to like in inner mitochondrial space. So what happens when that protons come through the help of F0 F1 ATP synthase, then it synthesizes ATP. That is how the oxygen is very much important in our body. Otherwise, there is no other role of oxygen in our body. Okay, there are rules for catalyzing some of the reaction for oxidation and other purposes, but the main role of oxygen is to accept the electron in the as a terminal acceptor of etc chain. And that completes the complete glucose oxidation, and we get the energy from it, and ATP is being synthesized. Now this ATP will be utilized for all over the all different functions in our body, whether it is protein formation, whether it is protein folding, or biochemical reactions, or glycolysis itself, which is the energy generator. Okay, or the fat degradation or formation, like, or the like various replication process. In replication also we need ATP. So whenever there is a for like breaking of bond, ATP will be necessary. So for all those process ATP is necessary, and we get the ATP as per our need due to the electron transport chain in during oxidative phosphorylation. So that we discussed in the enzyme mechanism also, like how hemoglobin and myoglobin kind of transport or store the oxygen in our body. Okay, so in this you ma'am also discussed lysozyme. Lysozyme and its mechanism, which is important. You have to read about it. Lysozyme and ribonuclease. What are the amino acids that are involved in the catalysis, and what is the substrate of this, and what is the substrate of lysozyme, which all amino acids are important for lysozyme functioning. In that I also showed how to operate during when I was discussing the lecture number 6. That time I also showed how to download the protein structure from PDB data bank, and then how to visualize that in the PyMol, and these all sessions I took during lecture week number 6. You can refer to the recording in the YouTube. Now we in week number 7, week number 6 we haven't talked about. So week number 6 was related to oxygen binding curve of myoglobin and hemoglobin, which is Hill's plot. What is the equation, which is very much important. Hill plot is very much important when you are understanding the Hill's equation. Hill's equation, hemoglobin and myoglobin binding curve. You must expect a question in the in this whether in this exam or in the competitive exam, whether it is CSIR-NET, DBT, or GATE Excel or GATE Biotechnology, anywhere you must have the questions from this. You will must have the question from lecture number 5 to lecture number 4 to 6. Okay, because this section is very much important in all kind of aspects. Then you also focus on protease catalysis. Protease is important. This topic is important. Why? Because when you have an entire protein and you want to analyze what are the different proteins, what are the different amino acid, and what is their sequence in that. So for that purpose, you need to understand which protease cleaves at which site. What is the cleavage site of the protein, or what is the cleavage site of that particular protease? What is the how it cleaves, or whether it cleaves at the C terminal, whether it cleaves at the N terminal, and important proteases like chymotrypsin, trypsin, and elastase, how they work. What are the amino acid involved in their catalysis? So after that, ma'am also talked about the Edmond degradation. So Edmond degradation is important technique to analyze the protein sequence of like amino acid sequence of the protein. Okay, during this lecture, week number 6, I also talked about I don't remember whether in the week 6 or before. In that, I talked about how once you have degraded the protein, then how you will analyze it using chromatography technique, or how a mass spectroscopy plays an important role. And maybe in the week number 3, I discussed what are the role, how X-ray crystallography is helpful in understanding the protein structure, 3D structure of the protein. Okay, then what is the role of BPG in case of hemoglobin regulation? This is very much important. This also I discussed in great detail in week number 6. So you can refer these before going to the exam, which is on 29th April. Okay, so now coming to the week 7. Week 7 was where is week 7? Yeah, in week 7 we discussed the lipids and membranes. So in lipids and membranes, what we discussed, the function of lipid, what is the glycerosphingolipids, what are cholesterol, how these are formed, what is the fluid mosaic model. Okay, so the important thing in the week 7 is, let me tell you the important point in week 7. Okay, in week 7, important point is transport proteins and channels, carrier, because you will get many questions from this topic. Channels, carrier, channels, transport protein. Then your fluid mosaic model is very basic one. You just go through it, and the another thing will be active and passive transport. Active and passive transport, and thermodynamics of membrane protein. Okay, so in week 7 you can focus on this. Now coming to the week 8. Week 8 was related. This is week 6. Okay, so week 8 was related to your nucleic acid formation, the definition of nucleic acid. What are the like in this you can focus on what are the various type of nucleotides, because whether you appear for interview or entrance examination, question from nucleotides might be there. Okay, then types of base pairing, you can expect numerical problems from here. Okay, the melting temperature, the DNA denaturation and renaturation, then what are the genetic code, what are the 3-letter codons of the 3-letter code to the protein, then hydrolysis of DNA using different endonucleases and exonucleases, and what are their functions. So types of DNA, then the characteristic of different DNA, Z DNA, A DNA, B DNA, what is the pitch length, then like how to do the base pairing, then you can focus on sense and anti-sense strand, because this will be important when you are studying the molecular biology also. This topic is very much important in case of molecular biology. So when you are studying the nucleic acid, during that time, like how this replication happens, how the transcription occurs in body, what are the enzymes involved. When you are studying about the complete transcription replication process, including enzyme and cofactors and everything, binders, so that becomes molecular biology. But when you are studying the chemistry of it, how it is being formed, then how it is how do you know that what kind of base pairing will be there, then how the helical structure will be formed. So these all things will be important in case of understanding the biochemistry and its thermodynamics. So I also discussed some of the numerical problem in this week. Okay, refer to the recording. Now in week 9, what we discussed? In week 9, we discussed about enzymes and like vitamins and coenzymes. Okay, vitamins and coenzymes. In that, we discussed what are the various enzyme types of vitamins. Types of vitamins. then cofactors, and your catalytic activity, then neurotransmitter, and other vitamins, and what are their roles, types of vitamins and their vitamin and their role. Okay. Then what are water soluble vitamin and lipid soluble vitamin? We studied that. Then uh, how cofactor that is associated with cofactor are nothing but like vitamins are some of the vitamins act as the cofactor of enzymes. So how this cofactor help in the like reaction mechanism by holding by holding its structure. So these all things we focused in week 9. Week 10 was related to carbohydrate, and carbohydrate, in that we discussed what are the different uh, in week 10 we discussed. This is week 10, right? Okay. In week 10 we discussed the various type of like sucrose, glucose, and what are the aldoketones, what are the aldose, what are the ketones, then small molecules such as sugar and monosaccharides, then epimer, then what are the chair conformation of this mutarotation, what is the mutarotation we discussed at a greater detail, then the linking of sugar or protein, esterification, amino acid derivatives, these all things we discussed in week 10. Okay, week 11 was again for the bioenergetics, and in this bioenergetics, well, what all we discussed in bioenergetics, we discussed the like outward transport of protons and ions, then energy coupling, then NAD and ATP formation. What is the role of NADH? What is the role of NAD plus? And what is the role of FAD, FADH2, how these are being found, what are the different complexes in electron transport chain, complex 1, complex 2, complex 3, and flow of electrons, how it goes from one to another complexes, what are the cofactors associated with all the complexes, then synthesis of ATP, how the motor works, and what is the catalytic site, what are the different form. So basically, there will be a numerical problem from this topic. And also, when you are focusing this topic, bioenergetics, you must remember that ETC, electron transport chain, is very much important. Okay. And we lecture number 12, which was the last one, we discussed our catabolism, anabolism, pathway, enzyme, their function, then what are the role of various enzymes in glycolysis pathway, what is fermentation and lactate is. I also talked about in greater details like how this enzyme how the pyruvate dehydrogenase complex is important, and why NAD plus is important in case of when there is an anaerobic bacteria metabolizing glucose, because it does not go into the Krebs cycle, so it has to regenerate NAD plus through running from pyruvate to lactate or ethanol, and which helps them to survive. Then we also talked about thymine pyrophosphate mechanism. Okay, so out of these all, whatever question you have, you can now ask any question related to it out of these topics. Wherever you have confusion, you can ask. Yes. Out of this topic, any doubt you have?
Sir,
Yes.
how to improve numerical question solving approach? Like
How to?
I not found easy to solve numericals in biochemistry. So how to
Okay.
tackle?
Numerical problems. Okay. So for numerical problems, like in biochemistry, you have you have to remember the important chemical formula. Okay, chemical formula means the equation, mathematical expression. Okay, so mathematical expression, the important mathematical expressions that are there in biochemistry, like that is related to thermodynamics. If we specifically talk about, so in thermodynamics, the important formula is how it is associated. The first thing you remember that delta G, the import, like you must remember what is delta G. Okay. So whether the formula is related to the standard free energy or normal delta G. So delta G is equal to delta G naught plus RT ln Q, where Q is your reaction coefficient. Okay, so reaction coefficient mean if any reaction is happening, A to like if any reaction is happening, like A plus B gives you C plus D, then reaction quotient or equilibrium constant, it can be ln Q or it can be ln K equilibrium. Okay, so Q will be here C concentration of product by concentration of reactant, C into D into A into B. Okay, so you put the reaction quotient, then you can calculate the free energy change. Okay, and for calculating the standard free energy, which is delta G naught, the equation is given as NF. What was the NFE? Okay, that is the potential. Okay, you can calculate this using NFE, the formula. Another thing that is there for delta G is equal to delta H minus delta E minus H delta T. Okay. Let me take few problems from the few problems from the assignment so that you can have one glimpse of solving how to solve these problems that we discussed in, I guess, whether it was there. In this, we did not have the question. Numerical problem was there in the week 9 is the vitamins. Nucleic acid, we had few numerical. Enzymes, protein structure, thermodynamics. First, we will discuss the thermodynamics problem, then transport chain problem. Okay. In thermodynamics, was there any problem? No. Membrane transport protein was there. So there was one. Let me go to the. Do you have any specific problem, or shall I give you a problem, numerical problem? Let me see if any the if any quiz assignment have this problem.
But I think enzyme part.
Enzyme part?
Yes, sir, enzyme.
So you have a problem with that enzyme like enzyme kinetics. How to solve the enzyme kinetics?
Yes, sir, numerical question. How to solve.
Okay. Numerical questions from enzyme kinetics. Okay. Then we will discuss that. Enzymes. Okay. So in enzymes, okay. So in enzyme kinetics, you can have the problems related to like Michaelis-Menten kinetics. Okay, so in normally Michaelis-Menten kinetics, what you have like that is related to velocity of the reaction. The equation is Vmax into S divided by Km plus S. Okay, and if you revert this this is your kinetics model. Okay, now in the next thing that you will have, if you reverse this, what you will have 1 by V is equal to Km plus S divided by Vmax into S, and this will give you 1 by V is equal to Km by Vmax into 1 by S plus 1 by Vmax, right? This is nothing but your Lineweaver plot, LV plot. So mostly the questions come from this thing. Okay. So the most of the questions that comes from this topic is you have to calculate Km or substrate S. So normally if you plot this equation, the equation you can plot by concentration of substrate and velocity of the reaction. So if you understand it, if you understand it, how this problem, like how this plot will be, this plot will be like if the Km concentration is much less than substrate concentration, if Km concentration is much less than substrate concentration, then it will be, the graph will move like this. Okay, the graph will move like this. Eventually it will reach to a velocity Vmax. Initially when substrate concentration is very less, very much less, substrate concentration is increasing in this direction, right? And substrate concentration is low in this direction because this is 0. Okay, when substrate concentration is very less, then that time this equation behaves in a first order kinetics. Okay, and when the substrate concentration is reaching to its saturation point, then the velocity is very high. Okay, then the velocity will be reaching to its Vmax. Okay, velocity will be this one. So in that case, it will follow zero order kinetics. Now, the first thing that you have to understand, if the question is given whether Km is greater than S. If if the substrate concentration is much greater than S, substrate concentration is much greater than Km, that means the reaction will proceed. If the reaction will proceed towards Vmax and velocity will be maximum in that case. Okay, so substrate will be much more converted. Let me take few problem from that. Whether this has the problem, any numerical problem. Okay, one problem is here. Let us solve this problem. The question is related to right now the substrate catalyzer reaction velocity of this one. Okay, when the concentration of substrate is 0.01 molar. Given as substrate concentration of substrate is 0.01 molar. And at that point, reaction velocity is V is equal to 20 micromole per minute. Okay, assuming that Michaelis-Menten constants are followed, what is the kinetics are followed? What is the reaction velocity when the substrate concentration is this and this? Okay, now the question is related to, in first part, it is asking what is the substrate when the substrate concentration is 1 into 10 raised to minus 5 molar, then what will be the reaction velocity? Okay, and in the second part, the question is related to when the substrate concentration is 1 into 10 raised to minus 6 molar, then what will be the reaction velocity? So how you will approach this problem? For this, the question is in the question itself. It is given that it will this reaction follows Michaelis-Menten kinetics. So Michaelis-Menten equation will be applied here. So V is equal to Vmax into S by Km plus S V is equal to Vmax. Vmax into S by Km plus S will be applied here. Right. So from here you can calculate Vmax and Km. Now the question itself, the Km is given. Km is equal to 1:10⁻⁵ molar. Km is given. From there you can calculate the Vmax. Right? You have substrate concentration. At this substrate concentration, reaction velocity is given. Km is given. So what all information you know? Substrate concentration you know. This substrate concentration you know. Km you know. V you know. What you have to calculate? Vmax. So calculate Vmax. How you will calculate Vmax? Reaction velocity into Km plus S is equal to Vmax divided by substrate concentration. This will be your reaction velocity, maximum reaction velocity. You calculate Vmax is equal to reaction velocity at this 0.01 molar will be 20 micromole, and keep everything in the same unit. All the answers are given in the micromole per minute, so keep this as a micromole per minute only, and change your units for the substrate concentration so that you don't have any problem while solving it. So this is 20 micromole per minute Vmax into Km. Km is 1.5 into 10⁻⁵, which is 0.1 micromolar. Right?
Yes.
This will be 0.1 micromolar. Okay. So because if you divide it by one more quantity, then it will become the micromoles. Okay. So 0.1 micromolar into, now here also the substrate concentration, the reaction velocity is given here. The substrate concentration is given here, 0.1 micromolar divided by Km divided by substrate concentration 0.1 molar. Okay. Now, when you understood that the substrate concentration is equal to Km. If you are the first thing that you should notice before solving this problem, the substrate concentration here is 0.01 molar, which is nothing but your how many like 0.01 molar. Okay. So 0.01 molar means this micromole. 0.01 molar. Now, first thing you should notice that without applying the equation itself, first thing that you should notice is Km is much lesser than the substrate concentration. Right? Or you can also say substrate concentration is much more than the Km value, because Km value is 0.05 molar. Right? And your substrate concentration is this. So this velocity will be, if you follow the equation, when the substrate concentration is very high, so in that case velocity will be Vmax. So whatever velocity it is following, this will be your Vmax only. So Vmax is equal to 20 micromole per minute. Okay. Are you getting my point?
Yes, sir.
So for first equation, by seeing itself, you can say Km you know, Vmax you know. So now the substrate concentration is compared to now S Km is equal to your S. So that means your reaction velocity will be V is equal to Vmax by 2. Why? Can somebody tell me why?
Because Km is the value of half of the Vmax.
Yes, because in the equation, if you observe, if in the plot, if you observe, this is Vmax and Km is defined as at the substrate concentration, because substrate concentration is the X plot, and here you will have Km. So Km is the substrate concentration when the velocity is half of its Vmax. So this will be Vmax by 2. That is why definition it is itself. And in the first question, it is asking that when the substrate concentration is 1 into 10 raised to minus 5 molar, 1 into 10 raised to 5, 10 raised to power minus 5 molar, then what will be the Vmax? What will be the velocity? Right? What will be the velocity? Now you already know your substrate concentration is at this concentration, the velocity is this one, and this concentration is very much greater than the Km value, because 0.01 molar is much greater than the 1 into 10 raised to power minus 5, which will be 0.0005, 0.0000345. This is the concentration of Km value, and 0.01 is very much high. So that means this the reaction velocity at this point will be is equal to Vmax. Now you also know the Vmax. Vmax is equal to 20 micromole per minute. Now you know Vmax. So in the first part, the velocity will be half of its Vmax, because your substrate concentration is equal to Km. So Vmax, your V velocity will be, velocity at this concentration will be Vmax by 2. Okay, which will be 20 micromole per minute divided by 2. That means 10 micromole per minute. Now coming to second part. In the second part, it was asked, section B, it was asked the substrate concentration is 1 is to 10 raised to power minus 6 molar. Then what will be the velocity? Now velocity will be substrate concentration into Vmax divided by Km plus S. Now your Vmax is 1 into 10 raised to power minus 5 molar multiplied by substrate concentration. Substrate concentration is 1 into 10 raised to minus 6 molar divided by 1 into 10 raised to power 5 molar plus substrate concentration 1 into 10 raised to power minus 6 molar. Calculate it, how much it will come? How much will it come? This is a micromole per minute. Which one is micromole per? Vmax is 20 micromole, sorry.
So all value are cancel out.
Yes? No, nothing will cancel out here.
Now sir, it's 10 micro
So,
per minute.
20 micromolar per minute into 10 raised to minus 5 divided by 10 raised to minus 5 plus 10 raised to minus 6. Okay.
Minus 11.
So it will be how much? This is 10 raised to minus 5
Minus 11.
plus 10 raised 1 10 into 10 raised to minus 5. 10 raised to minus 1. Sorry. This is your 0.1 into 10 raised to minus 5. Right? And this is your 10 raised to minus 6. Okay. So in that case, what will be your here? What will be, if you calculate this, how much you will be getting? 10 raised to minus 5, 10 raised to 5 cancel out, and then it will be divided by 1.1. 20 divided by 1.1. 20 divided by 11. So take a calculator. 20 divided by, it is 1.8. So answer is A. What is the answer? A. Right? Is it clear?
Yes, sir.
So you can, when you are approaching to solve this kind of problem, you first remember, first you notice what is the substrate concentration is given, and you try to solve for Vmax and Km. And then you can solve the related problem. Whatever substrate concentration is given, then you can calculate that. Okay. Other problem, if you have, enzyme kinetics is solved. Now, any other issues you have?
Hello.
Yes.
Yes, sir. I had doubt regarding chromatographic techniques.
Okay, chromatographic technique. Yeah, I will take that problem. Because chromatographic technique is right now out of scope of this biochemistry problem, but I will take that. Other than that, anyone have any related problem from the biochemistry?
Explain the formula, which one you are explaining first, like thermodynamics, enthalpy and entropy, and that one. Like,
Enthalpy and entropy.
yes, did
Okay.
free energy and standard free energy.
Okay. Okay. Yeah, I understood. And that was in, I guess, week number 7, right? Transport membrane protein. I will take one problem from here itself, and then I will see if you are solving this problem. Okay, this is related to the membrane differentiation. And how you will approach this problem? This problem you can approach by the formula which is given in the like here. See, the let us first read the problem. Whenever you are solving any numerical problem in biochemistry, you first read the problem 3 or 4 times. Okay, then only you will be able to understand the problem. Now in the diagram it is given. Now in the diagram it is given that this is a membrane. You have a semi-permeable membrane here. Okay, and in the one side, box 1 you have potential V1. Another box you have potential V2. And in both of the side, in both of the membrane, let us consider this is the outside the membrane. This is in like in the cell. If you understand it, this is inside corner and this is outside corner. Okay, so this is membrane. This is position 1. This is position 2. So now in V1 you have concentration of calcium ions is 1000 millimolar. Okay, and on the other side of the calcium ion, Ca2 plus concentration is 10 millimolar.
10.
Okay. Now, so what the question is asking, consider a cell membrane with a potential difference across the membrane is V1 and V2, and the potential given that the equation delta Q. What is delta Q? Delta Q is the chemical potential of this cell. So chemical potential it is given 9000 minus 9000 joule per mole. And you have to calculate the potential difference across the membrane. Matlab V2 minus V1 you have to calculate. What is the
Yeah.
V2 minus V1? And what all information you have given? You have been given gas constant R, which is 8.314 joule per Kelvin per mole. Rydberg constant joule per Kelvin per mole. You have given Faraday constant which is 96500 calorie per mole, and your temperature is 25 degrees Celsius. How to solve this problem? The first thing you should remember what is the mathematical formula, a mathematical expression to solve this problem. Now, you have been given a chemical potential and you have been given delta, you have been given RFT. Okay, so to solve this, what you have to know the delta G formula is delta G is equal to, the formula is given here in this sheet. Here, delta G is equal to RT ln your concentration of from one side to another side, because it is asking V2 minus V1. So that means it is asking the flow from this side to this side. Okay. So
Yes.
V2 minus V1 means C2, C concentration of calcium ion in second compartment divided by concentration of calcium ion in first compartment.
Sir,
Yes.
why we always take lower value upper side and larger?
No. There is no as such any convention. You like if the problem is given, like if A is flowing to B side, so B will be your product and A will be your reactant. So
Okay.
in this case, calcium 1 ion is transporting from one side to another side. Okay, so in that case, whatever it is given, so in whether you are writing quotient for your reaction, so in that case, you will write your Q is equal to B by A, product by reactant, or equilibrium constant. If you are writing equilibrium constant also, A plus B is equal to A C plus D, then you will write K equilibrium is equal to reactant product by reactant. So similarly, you have to write the in by out. Okay, तो यहाँ पर क्या इसका फॉर्मूला होता है RT ln C in by C out. Okay, plus ZF delta psi. Here delta psi is nothing but delta mu. Psi is your change in psi is your, here you have to write your chemical potential, or if psi is not given, then you have to write V2 minus V1. Your potential change in potential, which is you have to calculate. And here chemical, this potential is nothing but your delta G. Okay. Now, what all information you have already there? Delta G you have,
energy.
calcium ion you have, out you have, R you have, T you have, Faraday constant you have. What is Z? Can somebody tell me what is Z?
Have number.
Z is your charge number, charge on the ion. If it is K plus ion, then your charge is Z will be 1. If your Na plus ion, then charge will be 1 again. How much charge it has? If it is Mg2 plus ion, then Z will be 2.
That's 2.
If it is chlorine minus 1 ion, then Z will be again 2. So right now here you have in calcium, how much charge you have? 2 plus. So Z will be 2 plus 2, right?
So,
so what all information you have? You have here delta G given 9,000 joule per mole is equal to RT. R is given 8.314 joule per Kelvin per mole, right? Joule per Kelvin per mole. Then temperature you have been given in degree Celsius, but here remember what is the in the formula, what is the symbol, what is the unit here used for the Kelvin, for temperature, that is Kelvin. So you have to convert your temperature to Kelvin, and for conversion you have to add 273.15, right? And that will be how much? 298 Kelvin. 25 plus 273, how much it will? It will be 298 Kelvin. So multiplied by 298, multiplied by 298 Kelvin, then multiplied by ln. Now you have to understand which potential you are taking. If you are taking V2 minus V1, this is your V2, this is your V1, and in the reaction you have minus sign here. If you are writing in by and this is your out, right? So your C2 Ca2 in will be V1, this one. Okay, so that will be your 10 millimolar divided by 1000 millimolar. Okay, plus Z. Z is 2 multiplied by Faraday constant. Now remember, Faraday constant is given in the calorie per mole. So you have to convert this calorie per mole to Faraday constant. This is charge actually. This is not calorie. This is a charge constant. So, and charge will be cancelled by this 96500 multiplied by your V2 by V2 minus V1. Now you can calculate V2 minus V1 is equal to 9000. This is minus sign. Minus 9000 joule per mole divided by minus 8.314 multiplied by 298. Kelvin cancel. So it is joule per mole again. Now your 29 multiplied by 298 divided into ln 10 by 1000 divided by 2 into 96500. You calculate this and then you will get an answer of this problem. Okay, so this is how you get the point. So your answer will come around minus 12.5 millivolt or something. Millivolt or volt. What was the answer? I guess the answer given here was wrong or right. Have anyone solved this problem? The concept is like this only. Is it clear?
Yes, sir. Thank you, sir. It's clear.
Okay. So, and other problems, if you want to solve the delta G, delta H problem that was there in the which one, I guess? In which module it was? Potential, that is there in the thermodynamics, lecture 3. In the protein thermodynamics, right now I don't have any problem ready with me. Okay, so normally the formulas are I'm seeing if any problems are there. Okay, I think there are. Other than that, I will tell you the formula thermodynamic equations. See, normally when you are solving the thermodynamics problem, protein thermodynamics problem, so you have to remember few formulas. One is delta G is equal to minus RT ln K equilibrium, where K equilibrium will be product by reactants. But if you are taking the reverse of it, means if you in ln K, if you take reactants by product, then this minus sign will be plus sign. Okay, this minus sign will be lost. This is your delta G. Now, if you like this is your this is not your standard free energy. This is other than that, for a potential cell, if you have given a potential cell potential, so calculating that cell potential delta G will be NFE, where N is the number of charge ions, F is the Faraday constant, E is the potential gradient. Or you can write this E also as V2 minus V1, cell potential. Another formula will be delta G, the relation between delta G, delta H, and delta S. That where S is enthalpy, change in enthalpy. S is change in entropy. Entropy is the measure of disorderness. Delta G is change in
Free energy.
gives free energy. Okay, so relation will be delta G is equal to delta H minus T delta S. Okay, this is another formula. One formula, second formula, third formula, fourth formula you can have RTL and K. I have already told you. Then another thing that you can remember is activation energy formula. Okay, A is equal to, and that is your okay, for EA. What was the relation name? So here, the first thing you can get it from this equation also. And if you want to calculate, the like if how many molecules are colliding with each other and what are the reactions and product reactants. So in that case, the K, which is the rate constant of a reaction, will be pre-exponential factor A. A is your Arrhenius constant, A into e to the power minus EA by RT. Okay. Now A is pre-exponential factor. This is your rate constant of a reaction. EA is your activation energy. R is your gas constant and T is your temperature. Okay, so how do you use this formula? You use this formula by taking log. So that will be ln K is equal to ln A minus EA by RT. And what kind of problems are being asked from this equation is you have to solve a problem like a, let me show you any problem. If in a given equation, if in a given problem, if you have been asked that there is a chemical equation which is going from A to B. Okay, chemical equation in which A is the reactant, B is the product, and the activation energy of this to achieve when the enzyme is present, when the enzyme or any catalyst is present which is catalyzing A to B reaction, then and if it is given that activation energy is given as so and so, some value is given there, activation energy, and you change the temperature, you change the temperature of this reaction, then what will be the, and this activation energy is given is the temperature of 25 degrees Celsius. Okay, and pre-exponential factor is given as something x. Okay, then the question, if the question is asked, if you change the temperature to 75 degrees Celsius, then what will be the activation energy? Whether it will reduce or increase. Okay, so how to calculate that? First, you need to like calculate the K2 and K1, means rate constant at different reaction. If you know the rate constant at different reaction, then you can apply this formula for K1 and K2. ln K2 by K1 is equal to minus EA by R into 1 by T2 minus 1 by T1. Temperature 2 you know, temperature 1 you know, activation energy you can calculate through this formula if you know K2 and K1. Okay, and how this formula is you are getting from this to this, that you can get from ln K2. So if you fill K1 here, ln K1 is equal to ln A minus, because A will not change, activation energy will change, E1 by R T1. Okay, E will not change, activation energy will not change. Temperature is changing, so temperature has the significant role in rate constant. Then ln K2 will be ln A minus EA by RT1, RT2. Then you subtract both of these, equation 1 and equation 2, you subtract both of these, ln K2 minus ln K1 is equal to ln A minus ln A minus EA by RT1 minus EA by RT2. So when ln is in the minus sign, then ln will be K2 by K1 is equal to ln A cancelled out, and this will be EA by R minus, this is T2 minus T1. EA by R, then 1 by T2 minus 1 by T1. So by this formula, you can calculate what is the change in rate constant due to change in temperature. Okay, and these formulas you can use when you are calculating the thermodynamics of any given equation. Is it clear?
Oh, yes, sir. Sir, why we called like delta G naught standard free energy? Because it's happen in a standard condition, like
Standard condition means normally delta G naught is calculated as the temperature of 25 degrees Celsius. Delta G, it is called standard free energy because it is calculated at 25 degrees Celsius. So, and you calculate this with the help of this formula, because now temperature is changing, so delta G will change. If temperature is changing, so free energy will change. Why free energy will change? Because equilibrium will shift. So to calculate at a particular temperature, you have to calculate delta G at that particular temperature, and that you can calculate using delta G is equal to delta G naught plus RT ln K equilibrium. Okay, this will be your calculation of delta G at any temperature. Delta G at any temperature will be equal to delta G at standard free energy plus RT ln Q, KQ equilibrium.
Okay, sir. That means कि delta G naught standard free energy is at a specific temperature, like 25 degrees.
Yes.
Difference, na, sir?
Yes. Delta G naught is at a specific temperature, but if you are asked to calculate at different temperatures, then you use this formula to calculate the delta G at different temperature. Okay.
Yes, sir. Understood, sir.
Okay. Yeah. So what was you asked Tanya? You asked about what is the role of how do you calculate the chromatography? What was your question? Can you repeat? I will ask you. I will take your question now.
Yeah, so the doubt was that in chromatographic technique, based on what we want to separate, we select what the pH of the solution should be, of the solution we add as a buffer, right? Then the ionic strength and those different aspects of it.
Okay. So basically, normal chromatography has very different. In chromatography itself, there are various different chromatographic technique. Okay, there is paper chromatography, there is column chromatography. Then column chromatography. Then ion exchange chromatography. In column chromatography itself, you have various category. Ion exchange chromatography, what you were asking, that is related to your column chromatography. You have to change the ionic strength, you have to change the various pH, temperature, and these all things, like whether your stationary phase and mobile phase, like that. Then another thing is chromatography at high pressure. So that is HPLC and GC, gas chromatography. These are also the chromatographic technique. Now, to understand the chromatography, I will tell you in very brief sense. The first thing is you should have a stationary phase. You should have a mobile phase. Another thing, your sample or analyte. Okay, so these three things are basic, fundamental things required to run chromatography. Okay, these are the requirements. Now, the other things, which is pH, ionic strength, and other parameters, that changes pH, ionic strength, or nature of your stationary phase or mobile phase. That depends on what you want to separate out. Okay. And this principle is in all kind of chromatography. let us say you are running paper chromatography. In paper chromatography, what you take a paper which act as a stationary phase. Then in the chamber you keep a water molecule, not your mobile phase, and you keep this paper in this mobile phase, and then you write is you keep a sample such that you keep a dot of sample such that okay, you keep your sample somewhere here which it comes in contact with your mobile phase. That should be much deep in, or whether not deep inside the this is must be somewhere here, just above this line, above the surface of this water. Now what happens due to the capillary action? Due to the capillary action, this water or mobile phase, whatever liquid mobile phase you have, that climb above this. Hello. Am I audible?
Yes, sir, you are audible.
Yeah. Now,
Yes,
due to
sir.
the capillary action, due to the capillary action, the analytes that are there in the sample, they interact with mobile phase and climbs separate out throughout this paper. And the same principle applies in other kind of chromatography also. Now, in this sample, if in this sample you have A plus B plus C, both having all three having the different interaction of the mobile phase selected here. If they have the different interaction with the other analyte, so they will climb on the paper according to that, and similarly it will separate out. They will spread all through the column, and then by taking the ratio of how much the sample has risen and how much mobile phase has risen, you take the ratio and then you calculate the retention factor, RF, retention factor of that sample. Similarly, in case of your column chromatography, how you do? You prepare your column, which is your a tube, glass tube. There you have a kind of stopping mechanism, and you fill this with a stationary phase, which may be silica or any other material, which have your different interaction with the solvent. Let us say why you want to run this mobile phase. Let us say you have extracted proteins from a protein extract from a cell. Okay, from a bacterial cell. You had a bacteria. You disrupted the cell membrane and then you made a protein extract. Now this bacteria will have many proteins in its cells, and this was digested by proteases. Now in this sample you have lot many proteins, but you want to extract out your protein of interest. Which is, let us say, which is X. Okay. Now, and there are numerous other proteins in this extract, right? In this extract, you have numerous other proteins other than X. But you want to you want to separate this X from this crude extract. So you will kind of prepare your stationary phase such that it will have minimal interaction with, or based on the interaction parameter you decide what kind of stationary phase you want. So for this can be in the two approach. It can be either mobile phase interact with this. Hello. Hello.
Yes, sir. Hello.
again I'm facing some network connectivity issue. So I will wrap up this speed. So the first thing that you should focus while separating out your sample using column chromatography or any chromatography technique. There is some kind of interaction between your uh, solid state or stationary phase with your sample. Okay, for what you will think whether your mobile phase is interacting with this a lot or your stationary phase is interacting. So in first phase, what you will do, you will design your stationary phase such that your analyte of interest or your protein of interest will bind to these stationary phase much more stronger than the other proteins. Then what you will do after that, and that you do like how first you prepare the column with stationary phase, then your sample you do with a mobile phase. You run your sample with mobile phase, which is a liquid of less interactive fluid, which does not interact with these uh, as much this particular substrate or analyte is interacting with the stationary phase. Now you mixed your mobile phase with this one, you put your sample, and then you run the mobile phase. Now what will happen when this has the mixture of samples that is running through this stationary phase. In some of them you have your protein of interest, then this protein of interest will interact with stationary phase, and other green particle will run through the mobile phase and will come out first. Okay, then you collect it. Once you have collected all the your other component, then you run another mobile phase. You change your mobile phase, which have high interaction with these protein of interest. Now what will happen? This mobile phase, this protein will interact with the mobile phase, and this the interaction with stationary phase will loosen up, and in the next sample container you can collect this your protein of interest, and you subject this with the mass spectroscopy or fluorescence spectroscopy or UV somewhere, that whether your protein of interest is separated out or not, how much protein it has or how much compound it has, whatever you were separating out through the column chromatography. So because of the separating out, you need to know what are the interaction, what is the pH, because if you don't know the pH of your protein, in which pH it will be kind of stable, or in which ionic strength it should have, which ionic strength the mobile phase should have, so that it will interact in the second time and it will like uh, exclude out from the column. So until unless you know all these information, you won't be able to separate out your protein of interest or your compound of interest in general. So that is why these all parameters are essential in designing your chromatography experiment. Is it clear now, Tanya?
Sir, but they use the buffer like in anion exchange chromatography. They use the buffer which is having the positive ions. So that cannot happen because in anion exchange chromatography, you want that your anion gets attracted to the positive beads that are present in the chromatographic
Yes.
column. But if if it is already the anion is already dissolved in the positive ions of buffer solution, then it won't be able to attract to the beads which are positively
See,
charged.
yeah.
Why
So
do
that is why you change the mobile phase in second run. In first phase what you do, in anion exchange chromatography, the principle of anion exchange chromatography is like you have to select your buffers according to your ions that you want to separate. Okay, let us say your stationary phase is your stationary phase is positively charged. Then what kind of illusion buffer you need? What kind of illusion buffer you will need? If your stationary phase is positive charge, and through positive charge, stationary phase of positive charge, which kind of ions you will separate? Tell me.
Negative ions.
Negative ions, right? Your samples should have negative ions. So in the first phase, it will interact with the negative ions, right? Then whatever the other positive charge, there will be positive repulsion. So other who has less interaction, they will elute first, right? The unwanted ions, unwanted molecules, they will elute first. Now this is being interacted in ion exchange chromatography. This is being interacted first. Your sample which is having negative charge of your interest, it will interact with the stationary phase in first phase, and when the complete unwanted molecules have been separated out, what you will do now, you will change the ions that are there in the buffer. Okay, so that the highly interactive ions, because this is also negative charge, so to replace ion exchange means now in the second run you have to give some other samples in your buffer, some other analytes, not analyte, some other ions in your buffer, so that it can replace this, which is having the higher energy with this molecules which are there in the stationary phase. So you will, in the ion exchange, in the second run, you will put again a negatively charged compound in your buffer, so that your sample will come out, and which is having the higher affinity to the stationary phase, they will interact, and this will elute out in the second phase. Is it clear now?
So that means that the stationary phase, which has a positive charge, is much stronger than the positive charge of the buffer that we are using. That's why the negatively charged protein is being attracted to the positive charged beads in the column.
Yes. See,
Okay.
if in the first run, if in the first run your buffer having positively charged ions other than your solute, hello. Yeah, I am facing again and again the network connectivity issue. Am I audible?
Yes, sir, you're audible.
So you got my point, Tanya?
Yes, sir, I got your
Hello.
point, but there is one sentence that is written that the solubility of protein at low ion concentration increases as the salt is added. That is salting in. So,
Salting in and salting out effect. That are the different. That is the different concept. Salting in and salting out. That is not related to the ion exchange chromatography.
yes, sir, that is different. Salting out is
Salt.
different, but if you can explain what that means.